philic or is referred to as hydrophilic. The reason being that that side has an ability to attract water. It has affinity for water. So we say that part is hydrophilic. And it has a long tail. And the long tail is made up of long hydrocarbon or fatty acid chain. And we know hydrocarbons or lipid are also non-polar solvents. The reason is they do not have affinity for water because water is a polar substance, so it doesn't attract water as any polar solvent or substance can attract water. Due to that, it repels water. That's why we call it hydrophobic. That means water hating, and that is the middle part we have here. So it's a phospholipid bilayer. Bilayer means two. So Seymour Jonathan Sanger and Gart Nicholson Leslie in 1852 sorry 1972 tried to bring about another world definition or a definition of the structure of the cell membrane. So they said the cell membrane is a fluid mosaic model. And by saying fluid mosaic, they say that the structure that we see here is not that rigid. It's a little bit flexible, making it fluid for substance to enter. Now, why is it that something can enter into water? Something can enter into the water because water is not that close together. Because, sorry, give me some time. Give me some few minutes. I want to make some simple question for you. So sorry, give me some 2 minutes. I want to try if I can record it on my phone here. Give me some 2 minutes. Sorry. Yeah, thank you very much. Now everything is all set, so thank you very much. Okay, so as I was, please, someone you have the microphone is on, and I think either your parent or someone is talking about football. Kindly mute yourself. When it's time for you to talk, you can do so, so we can hear you very well. So thank you very much and for giving me the time. So they said that the cell membrane is fluid. So I was talking about water. Why is it that something can enter water easily, but that same thing cannot enter a solid substance? Something can pass through gases, like you can throw your hand in the air and you feel less resistance, but you can't put your hand through your mattress or your shirt. The reason is they are compact, it's not fluid. That is why a hard substance cannot pass through. So water, anything can enter water. Most something that is denser than water can enter or pass through. That is how the cell membrane looks like. From the surface, it looks like something very rigid that nothing can pass through. But since it's semi-permeable, it allows things to pass. So it has this fluidity that we call the fluid mosaic model that gives it the ability to allow something to enter. So we said it allows some things to enter unaided, while some enter with an aid and some are not allowed to enter at all. I explained this time last week that sometimes you can go to your father's room anytime, any day, or you can enter your own room anytime, any day. It is your house, it's your room. If your friend want to come into your house or your room, your friend has to go with you or upon your authority. That is something that can go aided. You can enter unaided. Your friend needs to be entered aided. But there are some caliber of people your parents or you yourself will not send to the house. These are the substances that will be not allowed by the cell to enter. So at least some of you don't know me. I am Sir Michael, and I teach some classes. I'm not the only teacher over there. We are four teaching in the Form 1, but currently I am taking them through this online platform. So for those who are seeing me for the first time, sorry, you don't know the one who is teaching. So maybe from time to time I'll off my screen so your attention will be on what I am teaching. Thank you very much, and I welcome you all. So that is the fluid mosaic model. So now let's focus on the slide. He was trying to tell us that the cell membrane is made up of a phospholipid bilayer. I've explained that recently, which is made up of embedded proteins and lipid. Now, let me explain the fluid. Fluid is something that can flow. So that talks about the fluidity in the cell membrane. And we can also talk about the mosaic. So normally in class, I used to tell you that you did something we call a paper mache in visual art in the junior high school, whereby you take papers, you recycle papers from different sources, put them together in a water, it gets split into pieces, you put them together with a glue, and it looks something very, very nice. So mosaic refers to like different assorted things, different things put together. So the cell membrane being made up, look at the structure very well, being made up of phospholipid bilayer. You see it has some other things like a sandwich, some other things put on it, and these are the proteins that are embedded in the cell membrane. So it means giving the cell membrane the ability to move like a slow streaming movement. And sometimes you can put something on the surface of water, a water in a cup is stagnant, that's what we say. But it do have some slow movement. That is why when you put a shape or something on that water in a pan, you see that from time to time, that substance or particle move from one end of the bowl or container to the other side. That is how the cell membrane looks like. It looks very rigid and stable, but it has the ability to do a slow streaming movement. And that is why substance can move into. It's like moving through a cereal farm or where we have grass. You see the grass over there. When the wind blows, you see the thousands of the maize or any other cereal plant, they move side by side. That is how it looks like. For when we are far off, you think it's ready, it doesn't move. But when the wind blows, you see that those things move slowly. That is how the fluid mosaic model try to also tell us. So it suggests that the cell membrane is a dynamic fluid structure with a mosaic of embedded proteins and lipid. So these men try to give us a better understanding of the cell membrane, that a model of cell membrane or of a phospholipid bilayer with embedded proteins and lipid, which is able to move. So it has the ability to move slowly. That is the fluid mosaic model proposed by Seymour Jonathan Singer and Gart Leslie Nicholson. These are the two men in 1972. They tried to bring a deeper understanding of the cell membrane. So we talk about the function of the cell membrane. For the sake of time, we will not spend much time. We talked about this last week. I've given you the document, so this doesn't need much time. So because of the structure of the cell membrane, it's able to serve as a physical barrier, preventing substances from entering the cell. So I told you at that time, the cell tries to allow important substance to enter it, whilst disallowing bad substance from entering. So normally the good things are allowed to enter, the bad things are allowed not to enter. And also some good things produced, example are hormones, enzymes produced by some receptor, sorry, production site can be produced from one cell. They will be allowed to live, not because they are bad, but because they are needed by other target substances. So if you're having network issue, the person that just stopped, leave the group and join right afterwards. Let me check. Are we more than 100? If you're more than 100, others cannot join. Then I'll share the YouTube link with you. Let me check from the other side to see the number of students I have over there. So if you're having an issue, don't worry. Leave the group. But once you leave, quickly join so that no one doesn't take your seat. A time will come with more than 100 and okay, today we are 58 currently. So you can leave and join anytime, any soon. So that's what happens. If you can't hear my voice or you can't see my screen anytime, do what I just told you. Leave it and quickly you join back to the page. So let me go back to what I was sharing to you people. So I was talking about the cell membrane and I'm done with the fluid mosaic model and now I'm explaining to you the function of the cell membrane. So it serves as a selective permeable membrane which allows movement of substances in and out of the cell. It also serves as communication or signaling. So the cell membrane becomes like the skin. And remember, our skin is for touch sensitivity, also communication. If I touch you, it communicates something to you. It means a lot of things. And the way I will touch you can also communicate to you. So the cell membrane is like the skin of the cell. Based on that, it also communicates with neighboring cells. They signal each other. And this is very important when it comes to cell division. So I've explained this in class before to my students that cancer cells have an overriding of this signaling because they cannot have this interaction. They overproduce cells, and that will cause cancer. So that is the importance of signaling. If there's space, cells can divide continually to fill the space. But if there's no space and they communicate, there is no need for continuous or proliferous division of cell to cause cancer. So that is a function of the cell membrane and their communication with each other. Now, the plasma membrane also plays a vital role in anchoring the cytoskeleton to provide shape and structure to the cell. So normally I tell students that don't say that the cell membrane gives structure and what we call it and support to the cell, but rather is the cell that do so. But for you to be on the safer side, the cell membrane doesn't give direct structure to it or support to it, but it also gives support to cytoskeleton by serving as a base for cytoskeleton to hold on. Now, once they are hanged on, they are able to give the cell structure and shape. So it's not a direct importance of the cell membrane. It's naturally given to the cell wall. But in animal cells that don't have a cell wall, the cytoskeleton will do that. But they need the support of the cell membrane as a base to hang around. So that is the function of the cell membrane as we talked the other time. To that time, I also explained the adenosine triphosphate, and I told you that is the energy currency of the cell. When we come to Ghana, our currency is the Ghana cedi. US, they will have the US dollar. Then we have UK and some other European countries use their euro as their means of transaction. So when the cell is also using energy, it uses energy in the form of adenosine triphosphate. Adenosine triphosphate. And I explained to you that the phosphate here is a phosphorus group. Three means three of them. Then adenosine is from an adenine group, and this gives energy in the cell. So one of the phosphate group can be used for cellular activity. Now we have two molecules, which becomes diphosphate. So the tri becomes di. When we use one out of the two, it also someone has raised their hand. Francis Ma, don't worry, I'll call you later for you to ask your question. I'll call you very soon. Thanks for raising up your hand. So adenosine diphosphate, when we use another one, it becomes adenosine monophosphate. So it's like taking your lunch box to school and you're having three types of meal, one for breakfast, or okay, let me say one for lunch and the other for supper. But normally we have two for breakfast. Once when they get to school, I want to say that eat this one in the morning, first break, eat the second one, then second break, 12 o'clock or lunch, you eat the third one. That is how the adenosine triphosphate works in our body. Now, there are proteins which form pores in the membrane through which substances can pass. And these proteins are also referred to as the transmembrane proteins. So we were talking about the fluid mosaic model, which says that the cell membrane is made up of a phospholipid bilayer with embedded proteins and lipids, which has the ability to move slowly. So together they can move. Now, these are the embedded proteins. We have three types of proteins. We have the transmembrane proteins. These are membrane proteins that run across or they span across the cell membrane. So look on my screen here, where my mouse arrow head is. You see this is a membrane protein called transmembrane protein. Because it spans across the cell membrane, substances can pass through. So molecules that can be transmitted across the cell membrane with an aid, some of them can go through this. So we have co-transporters and the rest. Next time we can talk about them. So here they move through a transmembrane protein. There's another one here. It's called a protein channel or transport protein. This one is can also be a because it spans across the cell membrane. Some are peripheral, or we can call them surface protein. So example, the surface protein here. This peripheral protein is also a surface protein because peripheral is attached. It can also be detached. As this headset here, I can detach it. My web camera here, I can detach it. They are peripheral to my laptop or to my phone. So we also have what we call the surface proteins and this peripheral. So together we have embedded proteins, Embedded proteins, that's what I'm showing from the middle here. So now look at my mouse arrowhead or my mouse pointer. What I'm pointing at is an integral protein. An integral protein means something that is like part of it. It's like it has become embedded in it. So integral protein are also called embedded proteins. So we have integral proteins or embedded protein. We have transmembrane proteins at the second one. Then the third one, we have what we call peripheral or surface proteins. And you can see here at your own free time, we have glycolipid, that is a glucose unit or a glycose unit attached to a lipid. We have globular protein. These are also types of protein attached. And because this one is at the surface, is a surface protein or a peripheral protein, but specifically called globular protein. Globular protein is a solid protein. We have proteins, some of them are liquid, some serve as enzymes. So any globe, that's like let me say irregularly shaped protein can be called a globular protein. Then we have carbohydrate groups on the cell membrane. Some of these are receptor site for hormones. Some of them too are enzymes that facilitate chemical and enzymatic reaction in the cell. I will later ask for a few questions. Now, some function of the membrane proteins. Now, the membrane proteins that we are seeing, let's look at their function. After that, I will open for questioning. So the membrane proteins that we see around the cell membrane, some of them are enzymes and they serve as for enzymatic reaction. So there are enzymes present in the membranes for chemical reactions which take place on the surface proteins, and these enzymes are needed in protein synthesis. So the other time someone says that the cell membrane facilitates protein synthesis. I said it's not necessarily so. The reason being that it's the nucleus that does it. The nucleus is the one that takes charge of all these things. But the good thing is the cell membrane will also have some enzymes that can play a role in the enzymatics, or let me say, in the enzymatic processes involved in protein synthesis, respiration, photosynthesis, and other metabolic activities in the cell. So the fact that the enzymes are there does not mean that it is controlling it. It's the nucleus that is giving the command. Like right now, I'm here, I'm the one doing the teaching, but I can instruct someone to be a co-administrator. The person can be muting your microphones, can be maybe editing people from the Google Meet or doing that. She is doing that because I have instructed him or her to do so. So the enzymes on the cell membrane are doing that. It doesn't mean the cell membrane is doing it, but the instruction, the control unit or the control room is coming from the nucleus. So that's also too much information on what the person said the other time. Now, some of these membranes also serve as receptor site. We call something key and lock when we come to hormones. So hormones operate on a key and lock. If I give you a key to open a lock, you must locate the actual lock that key can open. How do you do so? Sometimes you look at the name on the key and the padlock. If they matches, you just use, oh, that could probably be the key. If it's the same, you try them one after the other. This one fit. Looking at the whole, the way it is maybe articulated, this is the actual key that can open that lock. So enzymes and hormones also operate on a key and lock. So we say something that we learn about the characteristics of enzymes or proteins. Enzymes, so let me bring this one. All enzymes are proteins, but not all proteins are enzymes. Now, all of you are students, but not all of you might be students of Yas and Toa Girls. All of you are science students. That's what I presume. But not all of you will be in Science 3. So all Science 3 students are biology students or science students on this platform. But not all of you are in Science 3. Some of you might be in visual arts. Some of you are in home econs 3, 2, 1, and whatsoever. So all enzymes are proteins. But not all proteins are enzymes. Some proteins could be hormones, and sometimes fats can be joined, glycolipid could be hormones. Protein can be joined with lipids and they are hormones. Hormone like cortisol is a fat. So later we can tell much about that. So they serve as a receptor site. So these enzymes will be where the hormones can be activated or where the hormones could be accepted for them to play their role. So they also receive the so they are like what we call a protocol team, or let me say a welcoming team that will try to welcome the hormones for them to facilitate the function that they are supposed to play. So that's the function of proteins on the cell membrane. The third one, they are protein which act as skeleton for the membrane. And what they do is they try to support. So cytoskeleton, fibrins, and the rest, they are made up of what we call it proteins. So proteins like the structural unit of our body, basically. So that is the function of this membrane protein. Now, number four, they are proteins which actively transport materials across the membrane, and those are called the transmembrane proteins that I showed up there to you. And they are also being transported by using what we call ATP. Very soon, if not today, tomorrow, sorry, Tuesday, God willing, we will talk about the transport of substance in and out of the cell. We will talk about the two main types, that is the passive transport and active transport. So today, if you don't have a lot of questions, we can move closely to that. Then we can understand why we are using ATP or energy in movement of substances. When we are using energy, we refer to that as active transport. My fifth point, then I'll open for maybe 5 or 10 minutes questions, then I continue. Now, there are proteins which form pores in the membrane through which substances can pass. An example of them is the protein channels. So some will be a membrane that will run across the cell membrane. They are transmembrane protein. Some will also form pores or channels, and we call them protein channels. And they are also transporters for substances in and out of the cell. So as it stands now, we've talked about the cell membrane, the structure of the cell membrane, the fluid mosaic model, function of the cell membrane, and also importance or function of membrane proteins. At this point, if you have a question, raise up your hand. I will check those who have raised up your hand and I'll call you to ask your question. We have 5 minutes for this, so hurry up. Once I call you, make sure your microphone is unmuted. Okay, Nkunimdi, I've seen your hand up. Yes. Nkunimdi, Integra, sorry, Integra. Yes, please. Yeah, you are welcome. Integral proteins, not internal. Thank you very much. Right there, Osei. Now, after you lower your hand, or I'll lower it for you. Any other question? Hurry up. So, if there's none, when it's 8:15, I will stop the questioning. Okay, Ali do Mariam, open up your microphone and ask your question. Mariam, once you're done with the question, you can leave the page. Mariam, your hand is up. If you are talking and I can't hear you, it means your microphone is still muted. Or mute your microphone and talk to us. 8:15, I'll continue the lesson. So hurry up, Mariam. Mariam is not talking. Anyone else who have a question, raise up your hand. I'll call you. So you check where your mic. Yes, Francis Bar. Answer your question. Francis Bar. Okay, talk to us. Yeah, thank you very much. Thank you very much. We will talk about this the other time. Now check my screen. So plasma membrane is the same as the cell membrane. So example, there's a membrane that surrounds the vacuole. We call it tonoplast. It's the same as the vacuolar membrane. So it's another name for it. So now check my screen. You can see something here. Later, at first it was called, I think, plasma membrane also. There was something here that I talked to you about that time. Very good. So they said that, read from this point. A few years later, the discovery of a cell under a microscope. A living cell was seen to be externally covered by a thin, transparent, elastic, regenerative, and selective semi-permeable membrane called plasma membrane. Later, the term cell membrane was given by C. Nageli and C. Kramer. And I was trying to get the name C, but I think I have forgotten. Once I get it, I will give it to you guys. So that is the name. I think it was here. It's Carl. The name is Carl Nageli. But the C Kramer, I've forgotten the meaning of the C. So it's the same thing, cell membrane or plasma membrane. We have a membrane that surrounds the vacuole. It's also called tonoplast, and we have nuclear membrane or nuclear envelope. So someone will say nuclear envelope, someone will say nuclear membrane. It's the same thing. Thank you very much, Samuel Samson. I don't know. Sorry if I've missed the name. Now, Alidu Mariam, you can ask your question if you are ready now. Juliana Sima, yes, I think Mariam is not ready. Maybe she doesn't know whether she has raised her hand. Juliana Sima, unmute your microphone and ask your question. Yeah, talk please. The what? Capsid. No. I want to be sure whether it's capsid you are talking about. Okay, very good. Capsaid is not the same as lipid. Lipid, you know, is a hydrocarbon. That is a general name for fatty acid and glycerol. When we say capsaid, so when we talk about capsid is the outer protein shell of a virus. So virus, they also have their cell wall. So the cell wall of virus is called capsid, whilst the lipid, as explained earlier on, is the general name for fatty acid. There's no question on what I taught you. Let's continue. You can keep your question for the next session if I ask you to ask your question. So that's the cell membrane. We talk about the structure of it and the fluid mosaic model. Now we are coming to do this activity. So I'm doing this with you. Once you are ready, unmute your microphone, raise up your hand, then you answer question. Now the activity is, now read with me, but silently in your mind, though, so we don't bring a lot of echoes here. It says, study carefully the diagrams below and use it to answer the questions. So you can study it on my screen. Let me mute it. Sorry, let me minimize so you can see all of them together. But at least you know the animal and the plant cell already. So this one shouldn't be a serious thing with you, ladies and gentlemen here. So now we are studying the animal cell and the plant cell. What is the main difference between the outermost layer of the plant and animal cell? I will just pick one answer. So someone should raise up your hand or mute your microphone and tell us what is the main difference between the outermost layer. A hand is up, but I've not seen the hand. If your hand is up, unmute your mic and tell us. Esther Kumache, your hand is up. Esther. Okay. Simple as that. Thank you very much, Esther Kumache. Any other hand? So after that, you mute your microphone for us. So after talking, just mute the microphone so that the feed will not be too much. Thank you very much, Esther. Okay, so anyone else? I think okay, that answers it. So I'm sure anyone else will say the same thing. Let's continue. Now, identify any cells. So when you come, just tell me two or one. Identify any cell structures or organelles in the animal cell and also plant cell. So let okay, this one the one person when you come mention six. Mention six. Okay, Olivia Free. Hey Olivia, okay. A champion. Olivia, talk to us. Mention six organelles in the animal cell. Mention all the six because of time. Olivia, unmute your mic and talk to us. Okay. Okay. Mm-hmm. Okay. Very good. So when last time we talked about organelles. So organelles are specialized structures in the cell that perform specific function. So they are specialized structures or they are structures in the cell and they perform specific function. There's a theory that says that even mitochondria, later I'm sure we will study it in our syllabus and in class, that they assume that some mitochondria was a bacteria on its own that was able to, okay, let's talk about first the chloroplast. The chloroplast was a bacteria on its own that has a chlorophyll, was able to produce its own food. It was later being surrounded or engulfed by a larger cell and become part of it. There are theories, they might not be entirely true. I don't believe most of these things, but that's a theory from scientists. And the mitochondria too it was a non-photosynthetic organism or structure, bacteria, that wasn't able to produce its own energy, but rather it was using respiration to produce energy. It was also engulfed by a bigger cell. These are all part of the cell theory. So that is the idea. Shanita Jifaka, tell us the next answer. Structures in the plant cell. Mention six to us. Shanita Ka. Once your hand is raised, be on your screen. We are now 62, so at least we can still use the Google Meet. Next time, I will share the link with you for the YouTube. Shanita, your hand is up. Unmute your mic and talk to us. Okay, Harriet also take the spot. Let me lower Shanita's hand. She is not aware the hand is up. Harriet also attend. Six organelles in the plant cell.
Ribosome,
Ribosome, okay.
nucleus,
Nucleus.
mitochondria,
Mm-hmm.
Golgi apparatus,
Wonderful. What again?
rough endoplasmic
Reticulum. Okay, so here it said endoplasmic reticulum, but you did well by specifying them. We have the rough and smooth. And why is it rough? Because it has ribosome on them. So the rough endoplasmic reticulum has ribosomes on their surfaces, and the ribosome are also used in cell, protein synthesis. So that is the function of ribosome specifically in the living cell. Harry, thank you very much. Let's look at the next question. So you can try this later on. It's a test you can do on yourself. Once I've sent to you the documents, feel free to read through anytime, any day, so that once I come here, you understand. You bring critical and let me say learned or literate questions, so that at least you understand better. If you don't read, you ask questions that you expose yourself or you yourself should know. But once you read, you can add educated and let me say guided questions for better understanding. Now, which three organelles in the animal cell is bounded by a double membrane? So we say that the cell is bounded by a membrane. There are also organelles in the cell that are membrane bounded. But here we're looking at that are double membrane bounded. Someone to help us with some of these structures. If your hand is up, lower your hand and talk to us. I want to monitor. Yeah.
nucleus and mitochondria.
Nucleus and mitochondria. Very good. Nucleus is there, mitochondria is there. Very good. Thank you very much. Any other structure that is also double membrane bounded? That is not all. She said too. She's really done very well. Any other person to help us? So people want to join whilst we are already on the platform. Let me see if they can join our meeting here. Okay, please, if your hand is up, I've left the page, so I can't see whether your hand is up. If your hand is up sincerely, just open your mic and talk to us. I'm on the WhatsApp platform trying to add some people. The late come.
Endoplasmic reticulum.
Very good. Has someone talk about the cytoplasm? If no one, sorry, that is okay. Sorry, that's chloroplast. That is what I wanted to mean. So chloroplast is also part of it. So these are all membrane bounded, and they are double membrane bounded. That and the reason is they perform special activity, and their activity has to be separated from the entire cell. So that's why some of these organelles also have their own membrane bound. Now let me come back to you on the Google Meet app. Now, describe by writing. This one, we can't do that here. It says describe by writing the shapes, structures, and function of any four structures of the cell. It's a question you can try on your own at your free time. Bambi Bell, your microphone is on. Let me mute it for you. Okay, so I've done that for Bambi Bell. Please make sure your microphone is always off. And that of I saw one person. Yes, you've done well. You've muted it on your own. Thank you very much. So now we have a clear sound, no feedback for any one of us here. So let's continue on. Now we are going to talk about movement of substances in and out of the cell. So by now, let's go to the PowerPoint. I did a very simple PowerPoint over there. Let me show the PowerPoint. Then once we are done with it, with this content, we can come back to our PowerPoint. Sorry, not PowerPoint, PDF slide. PDF slide. So now, uh, sorry, that was my former school, so I couldn't change it. So don't worry, it's the same information that we need. So I did it for another thing, and it's very important we can use it here. So by the end of this lesson, everyone should be able to explain the need of substances to move in and out of the cell. So why must substance move into the cell? Why must some also move out of the cell? There's a need. If they are all moving in, so we call something migration. We will later learn in ecology. So if people are just moving into Ghana, no one is leaving, something happens. Although it is bad that people die, but biologically it's good that people die. Sorry for saying that. Because if our great grandmothers are not dying, where will you stay? Now, some of us we are living in our uncle's room, a room that was given to our grandma. We are still over there. Should your grandmother be there? Would you have been there? I'm not saying it's good they die, but for life to continue, we call something carrying capacity. If it exceeds that limit, a lot of deaths will come. So later I'll explain that when you're talking about ecology. So it's good something's moving, something move out. Now, if you are old ladies, yes, old ladies, Yaoga and Momba and other school, they are old boys and girls. If they are still on campus, even the form twos, when you were in school, they were in the house. If they were to be there, would you have get a class to study? Now, Form 2 Science 3 has taken Form 1 Science 3 class, Form 2 Science 11 has taken Form 1 Science 11 class, and so forth and so on. So it means that there's a need for people to move in, people to move out for equilibrium and for stabilization in the ecosystem, also in your body, because we have an internal ecosystem whereby structures and substances have to move in and out of a cell for stability and smooth flowing of your body. So now we look at substances that can move in and out of a cell. Last time someone asked the question, I've forgotten, we realized that essential substances move into the cell, and I told you simply that unwanted or bad ones will leave the cell. Example is enzymes, hormones, sorry. Hormones are not normally needed by their site of production. They are transported. So a hormone can be secreted and it will leave the cell to another cell. It is living though. Now glucose can enter, water enter, enzymes also enter a cell. These are some of the important things that can enter a cell. We will talk about them very soon. Then we look at types of ways in which substance can move in and out of a cell, and that is what we call passive transport and active transport. Now let's move on. I've explained this already to you. That is the structure of the cell membrane. Now, the structure of the cell membrane we say is a phospholipid bilayer. This is the phospholipid unit, and this is the what we call sorry, this is a phosphate unit, and that is a lipid unit, the tail. Some on the microphone is on. Let me get the person and unmute it, please. Thank you very much. Oh, so sorry. Mamiba said I'm moving too fast. Yeah, I knew I was moving fast. So sorry. Let me slow down a little bit. But if I have said something that you don't understand, let me know. Naturally, I have adapted to fast speaking, and that is not the best for this. So sorry, I'll slow down. Some of my students are used to this, so sorry. So I was talking about the structure of the cell membrane, and those who couldn't hear that, we say it's a phospholipid bilayer. This is the phosphate group, and down here is the lipid group. And we said it has it's a fluid mosaic. So the fluid means it can flow. It is a little bit flexible. There's a flexibility and fluidity in it. So you can see here, something can just enter and go. Likewise, you see bush somewhere. Hey, this bush there, no one can pass through. But when you get close, you can find your way in the bush or the thistle over there. That is how the cell membrane looks like. And we said mosaic. Mosaic means assorted things, different things put together in one place, like a sandwich. As ladies, you know better than us. So we have different vegetables, or let me say, I'm not saying vegetables on a cell membrane, no. And this is just an illustration. So we have different protein molecules and lipid embedded in the cell membrane. That is the structure of the cell membrane. So this membrane is embedded. You see, it is inside it. It's an integral protein. This one too is embedded, and because of the hollow space, it's a protein channel that can help transport substances in and out of a cell. Now, this protein molecule, from where we can see it, it is at the top of it, so it doesn't move inside the lipid layer over there. So this one is a surface protein, or also called a peripheral protein. So that's the structure of the cell membrane, as I explained earlier on. Now, why must substances move into the cell? Some hands are up. Later, I will allow you to ask your questions. Vixie and the rest. Sorry, I will allow you. Now the question is, why must substance move into the cell? One, because of time, let me just, they are there, you've seen it. I would have asked you guys to tell me. So the first one is for cell to obtain nutrients after eating. That's why we said excretion is different from even going to toilet. Sorry, ingestion is not excretion. So when you go to the toilet, you have done ingestion, not excretion, because at that time the food that you are taking it out, the unwanted or undigested part, hasn't entered the cell. So they've not taken part in metabolic activities or cellular activities, and we say it is not excretion, because excretion is the waste product of substances that have entered the cell or has undergone metabolic activities. So now that you have eaten, the food hasn't entered the cells. It has to be assimilated through the ileum in the small intestines. Then by absorption, it moves to that surface cells, then into the blood. It can then be transported via the blood to all part of the cell. So for substances like food nutrients to enter the cell, that is why substances have to move in and out of a cell so that the food that you've eaten can enter your cell, the point of energy production to get ATP from the food. Because your body, your cell doesn't know carbohydrate, it doesn't know proteins and the rest. What it knows is ATP. If you have Ghanaian cedis in US and that store doesn't take Ghana cedis, you are like someone who doesn't have money. You have to go to the bureau to change it, or unless they decide to accept that you have money, that is when you can say you have money to buy something. You have a purchasing power. You have to convert to their currency. So that is why cells have to get nutrients into the mitochondria for them to undergo respiration, then reform energy in the form of ATP. Second point, after this slide, I'll allow for questions. So don't worry. If you have a question, focus on what I'm teaching. Note it down, focus on what I'm teaching, then you will come in. Second one, once we have entered nutrients to enter, waste will also come. So to also excrete waste substance. So I told you the good things will enter the cell and the bad things or what is not needed by the cell but needed by other cells, that is secretion, can also leave the cell. So we also excrete waste substances. Human cannot live without waste, we will die. Plants can keep their weight at the tannin, the resin, the rubber, and the rest. They can stay with them for so much a long time, sometimes for life, and the plant will not die. But humans, because of our metabolic rate and our waste not being converted to harmless substances, we have to take them out of our body, else we die. So for we not to die of starvation, we have to get nutrients into our cell. For we not to die out of waste accumulation, excess waste or waste substance have to leave our cells through excretion. Now, also to secrete useful substances. So example, I use the pancreas. We have a cells called the islet of Langerhans that produces two types of the beta and the alpha cells. The beta produces insulin and alpha produce glucagon. So once they produce these enzymes, sorry, hormones, the hormones have to leave the pancreas to the blood, where they will tell the what we call it, the hypothalamus that hey Charlie, tell the pituitary gland that there's a lot of carbohydrate, glucose in the blood. Let's convert them to glycogen so that the person doesn't get diabetes. So insulin will convert excess glucose into glycogen. Now, when there is low sugar, that's hypoglycemia, the same will be produced, rather the antagonistic hormone called glucagon will be produced. Then it will convert excess glycogen to glucose so that your lower glucose level that can make you faint or die as a result will now rise steadily. So that is the function of the hormones produced by the pancreatic cells. So when they are being produced, they don't have to stay there because they have a site where they can do whatever they are supposed to do. So they will live into the bloodstream where they get into the blood, they do their thing. So for we to secrete substances that are useful to other part of the body, there's a need for substance to move out of a cell. Once it moved out of that cell, it also enters different cell on the bloodstream. Then they do their work or their function. Also, to maintain a suitable pH and ionic concentration in the cell for enzyme activity. So these enzymes has to get the activation, and the activation is through the movement of substances. Sometimes it could be sodium, we call something the sodium potassium pump that helps in muscular activity. That's the fifth point over here. So also to generate ionic gradient or ionic potential for nervous and muscular activity. So for the cell or the nervous system to be able to do their function, they also need movement. So we call something the sodium potassium pump. When these enzymes, sorry, when these ions move, it causes what we call ionic potential. Now you learn in physics and chemistry that when there's a difference in potential differences or there's a potential difference, that is when electrons can move because one is higher, one is low. So there's a movement from the negative to the positive charge of the node. That cause that same thing happening in the cell. So a biochemical reaction occur that causes nerve impulses. So I can move my hand. How do you feel it? Because there was a inside movement of sodium, outside of calcium, causing sorry potassium, causing a potential difference, what we call ionic gradient, so that muscles or nerve impulses can transmit as if electrons are moving in your nerve. So that's what happens when substance move in and out of a cell. Without it, you will not feel or your nerve will become numb. You don't feel anything in your skin. Now, I'll pause here. If you have a question, raise up your hand, then I will explain shortly. I have asked you guys to make it 1 hour 30 minutes. Now I've told you by next 2 weeks or so, our math teacher will start teaching. God being so good, our physics master, Mr. Sampson, has also created a group just yesterday night. I invite all of you to join, and I'm sure once you join, you will need some days over there. So it means that he will have to take the other days for different times, or maybe one day, one of the biology days might be taken off. So once that we have time, let's utilize the 1 at 30 minutes. If they take one day off, we can still have enough time. So sorry, let's make it so 9 o'clock on the dot, we are done. I can go for 2 hours, 3 hours, but some of you are students by 9, you should be off your phone, prepare for the next day. So a hand is up. In Kunimde or Secretary, I think you are the first hand that I've seen here. You can ask your question. I was just having a commercial. Yeah, here they ask your question. Kunimde. We have what? Yeah, key and lock system. That is how hormones identify their receptor site. Receptor site. Oh, okay. It's a key and lock. Lock, like how you say key. Who's a key open a lock? Have you got it? Yeah, LOCK. So once they enter, so like assuming this is the key. Now look at me on the screen. This is the key. That is the lock. So you see that this one can fit into what I've done over here correctly. So if you bring something like this, it doesn't fit. So something like a circle has to fit into it. So this could be the lock. This could be the key. So when a hormone like this come, it cannot fit. So it will not act on this where it will be activated. But when the hormone like this come, you see a receptor side like this, it enter, it becomes activated, they bind. So we call it hormone activator binding or activation. So once they bind together, that is when the hormone becomes to work. So let me bring a simple example. Biology is very broad and I don't want to bring a lot of things. So it's something in a different topic. But since you have asked, let's make it simple. Remember, we have an enzyme for we call prorenin. Renin is for digesting of kid. Let me say milk in normally it's found in kids. Adults have it, but it's predominant in kids. When the hormone is being produced, it is called prom enzyme. Sorry, it's called prorenin. But upon activation with ACL, it becomes renin. Let me bring another one. We call something this enzyme. In the for the polypeptidase. Yeah, pepsin. Very good. Pepsin. Thank God I've remembered. It's called pepsin for digesting proteins. When it's being secreted, it is pepsinogen, and pepsinogen is inactive. But HCl will activate pepsinogen to pepsin. What happened? HCl has a binding site to the pepsinogen. Once it becomes activated. Also in blood, it is chysinogen, blood clotting. a carcinogen. When carcinogen is also released to the atmosphere, when it gets exposed to oxygen, it now becomes casein, and it becomes like something like a milk curd that will try to clot your blood. So most of these things are inactive. Like now, you are very quiet. Once I join one of your friends, you say, "Hey, hello, maybe Betty, how are you?" You see that you are talking. But once you are alone, it's just you. You are very quiet. But once you see your friends, at least you have an interaction. So that is why God has made them so that the hormone will not be working. They only work when they see their actual place they're supposed to work. So thanks for the question. Maybe later when you reach enzymes and these things, you will understand them better. But for the purpose of today, we can't show the diagrams and everything about it. But one day, if I had one slide like that, I might put it on the WhatsApp page for you to understand. Now the next question, I'm sure you are okay, our lady.
Yes,
Yeah, thank you very much. So the next person can ask your question. If your hand is up, or mute yourself and talk. I can't see those who hands are up. I have to go back. So if your hand is up, kindly or mute yourself and talk. If no hand is up, I will continue. We have few minutes to end our lesson. We are almost 1 hour into our lesson. So 1 hour 30 minutes, we are supposed to end it. Okay, I don't. Okay, Harriet, also your hand is up. Please talk to us, Harriet.
hello, sir.
Yeah, hello, Harriet. Welcome. Talk to us.
You said the rough endoplasmic reticulum has ribosomes.
Yes, attach on their surfaces.
Okay, so the ribosomes are the membrane found in the rough endoplasmic reticulum.
Sorry, I got lost. Ask the question again.
You see, he asked the question that some of the organelles have bound membranes. Sorry,
Yeah.
yes.
Yes, please. That's true.
So, and you said the rough endoplasmic reticulum has ribosomes.
Yeah.
So is the ribosome the membrane to the rough endoplasmic reticulum?
Okay, thank you very much. Stay on the line. Let me explain quickly. Because if not time, I would have asked one of your friends to even answer you. So the point is, this a ribosome is also an organelle on its own. And what we call the endoplasmic reticulum is another organelle. It's normally for transport. They help in packaging and distribution of lipid and other structures in the cell. Even when proteins are being produced by the ribosomes, they pass through the rough endoplasmic reticulum to the cell, and so it's like the system, the channel. So the cell membrane, the endoplasmic reticulum has its own membrane that surrounds it. That's endoplasmic reticulum membrane. And the ribosome has its own membrane. So the ribosome as an organelle, most of them are dispersed in the the cytoplasm. Some of them are dispersed in the cytoplasm. Some are found in the nucleus. And some are also embedded on the endoplasmic reticulum. So it's like they are spread across. Some are in the nucleus. That is where actually they are supposed to be. Some are also in the cytoplasm. But those in the cytoplasm, some are fortunate enough to have them attached to the endoplasmic reticulum. So they are not the membrane of the endoplasmic reticulum. They are other organelles being attached to another organelle. Are you okay, Harriet?
Yes.
Thank you very much. Oh, you are welcome. So lower your hand. During appear, talk to us. Once I mention your name, unmute your mic straight, you ask your question. We have less than 12 minutes to end the lesson today. Doreen appear. Are you the one talking, Doreen? Yeah, talk to us. Please, your background is a little bit noisy and your network is breaking.
Please, the substances that moves into the cell that is to get into an organelle before it works.
So if I get to your question, substances that enter the cell.
Inside
No, no, no.
that moves into the cell that needs to get into an organelle before.
So now let me ask you a question from your question to understand. After it has entered the cell, must it find itself in another cell to perform its function? Is that your question? Doreen. So your question is after it has entered the cell, must it enter another organ before it can perform its function? Yeah, Doreen, did you hear my
Yeah, that's
okay,
my
thank you very much. Unmute yourself because your environment is a little bit noisy. Next time you can find a secluded place then only you can study. So let me try to explain Doreen's question. It's a very, very good question. So now that the substance has entered the cell, now very good. You see that your room, I use this as an example. You have a very large house. So you have a living room, the dining hall area, a study room. If you have the living room. Okay, I'll talk about that. You have the kitchen, storeroom, and the rest. Now, when you bring foodstuffs, I'm sure Doreen, you don't leave it anywhere in the living room. You will take it to the storeroom or the kitchen. That is where you will use the food for preparation. So if something like carbohydrate, not even carbohydrate, something like glucose enters the cell, carbohydrate cannot enter the cell unless it's an enzyme or maybe a hormone substances. So if it's a food nutrient, glucose, when glucose enter the cell, where do you think it will be? Anyone should raise their hand and tell me. Glucose will not be in the cell anywhere in the cell. It will go somewhere. The one that raised the hand, just talk to me. Where do you have it? Where do you think glucose
mitochondria.
very good. It will move that to the mitochondria. That is where we have the mitochondria to break it down through respiration. So most of the substances will have to get to an organelle. Something like water. Water does not necessarily need to go into any organelle. Water can be in the cytoplasm. And even the cytoplasm is even an organelle. Sometimes you can refer the to an organelle. So it could be somewhere depending on the structure. Something like ribosome. Okay, well, ribosomes are organelle over there. So something like DNA or any structure, enzyme or protein that will be needed for cell cycle, or let me say, sorry, protein synthesis, it will enter the ribosome or the nucleus. That is where we undergo the what we call it the protein synthesis, replication, and the rest. So it's a very good question. Some substances will find themselves in some organelles based on what they will be used for, but some will just be in the cytoplasm and the cell will use them as a whole. Harriet, your hand is up. This is a question you can ask, contribution you can add it up.
So I wanted to ask if could be that two organelles in a cell will need a substance at the same time.
Thank you very much. So now, yes, for instance, water. Water is needed in the mitochondria. Cytoplasm also need water because cytoplasm is a jelly-like substance. I've forgotten. It's about 90% or maybe more than 70% water. The others are dissolved nutrients, salt, carbohydrates, and the rest. So yes, at a point in time, more than two organelles. Sometimes even the entire cell needs it. Because water, every part of the body needs water. Water will be needed in the chloroplast. Remember, we have what we call photolysis under the light phases of photosynthesis, whereby light will split hydrogen into H plus, it will split water into H plus and O minus. That is the H plus will be needed to do the what we call the carving cycle and the rest. So water is needed in the carbohydrate. So water is needed in the cytoplasm, also in the mitochondria for respiration. So water could be anywhere, anytime.
And so the substances relate to each other, or one will be used and then move to the other one.
Yeah, the cell they will just be absorbed. So let's assume water. Water will enter the cytoplasm. So now it's in the cytoplasm. Cytoplasm also needs it. What happens? Some will also be transported to the mitochondria because mitochondria is in the cytoplasm. Have you seen the point here? You see that all the other organelles are suspended in the cytoplasm. So if there's water in the cytoplasm, it will pick it water from the cytoplasm. Chloroplast, you also need water. What do you do? Since there's water inside the cytoplasm, you also pick your water from the cytoplasm. So the cytoplasm is like the blood of the human system. The blood contains oxygen, carbon dioxide. When they get to the actual site, it will offload the carbon dioxide or the oxygen for the cell. That if there's water, if the cell lacks water, once the blood flows there, the cell also get hydrated as a flow of the water around a cell membrane. So two or three organelles might need the same resources, but it is shared to them or they absorb. And we will talk about diffusion very soon. That will help them to absorb whatever thing that they want. Osmosis too is there to help water to move into these organelles at the time that they want it. Are you
Okay,
okay?
thank you.
Okay. You are welcome. So thanks for the question. In
Yes.
fact, you are asking very, very intelligent and good questions. I'm sure all of us are getting understanding. Is there any, okay, I think someone, okay, Gregoria, your hand is up. Later I will call the one joining with, okay, Patrick Denchi, that's our father's Reverend Patrick Denchi. So the lady you can talk to us. Gregoria, talk first, then our sister using Mr. Reverend Patrick Denchi's line can talk to us. Gregoria, once I mention your name, you have just 2 seconds to talk. We have less than 7 minutes to end the lesson. 9 o'clock, I want us to end it. So please, Gregoria, I'm calling for the last time. Or mute your mic and talk to us. Okay, Gregoria, sorry, let me lower your hand. When you are ready, come again. Okay, Patrick Denchi, our sister, help us talk. Ask your question.
Sir, please, the question Hayat asked, is it only water or any other liquid substance?
Okay, it depends on it. Water is a universal solvent. It can go anywhere. Something like glucose. When glucose enters the body, cytoplasm, you don't use glucose for anything. Glucose will be in the cytoplasm so that it can be transported into the mitochondria. Then mitochondria will convert it to ATP, adenosine triphosphate. Now it's like I bring safer. If I bring safer to Kumasi, how do I use the safer for? But where I am right now, Sampa, you can use safer. I can go to the bureau and change it. Cote d'Ivoire people can bring their safer here. They can buy things in Sampa and they can go because we have some bureau or people that can exchange the money for us. But when I take it to Kumasi, over there, most people don't even know safer unless they go to the bureau. So they have no need of it. So the point is, when you bring glucose to cytoplasm, it can't use glucose. Rather, it will keep it so that it will be transported to the mitochondria, and mitochondria will bring ATP. That is the local currency for energy in the cell. Then ATP, for ATP, all the cell will need it. Sorry, all the organelles will need it. It will be transported to any organelle for all the activities. So some substances will be needed by all. Example is ATP. All of them, water, all of them need it. But glucose, mitochondria will take it. It will split it into ATP. Then all the other, something like sodium, potassium pump, all of this will be needed. If it is a food for storage, vacuole will store it. For example, excess starch, excess glucose, vacuole will keep it. Instead of being the cytoplasm, it will now be absorbed by the vacuole, and vacuole will store their products for them. I hope you are okay.
Yes, sir.
And please, your name so that next time I can call you by name. What's your name?
Sir Nanama.
Okay, Nanama. So next time I see your father's line, I'll call you by Nanama. If you're okay, you can lower your hand. Doreen Apia, talk to us. Doreen Apia, you can talk to us.
Sir, please do the organelles need the help of the nucleus to work?
Very good. So let me ask you a question. Does your body need your brain to also function? That is true. Yes. The nucleus is like the CPU or the brain. Of the nucleus control a lot of things. Now, you might think that your brain doesn't control your breathing, but your brain controls the sympathetic and parasympathetic and every part of the nervous system. Though you will not stop, you might try to stop breathing on your own, but your sense of survival, your survival instinct in your brain will stop you from holding your breath when you're about choking to death. So the brain does a lot of things, control things, whether you are asleep, everything, most of them, your brain and your nervous system does it. That's the same thing as the nucleus. The nucleus is the control mechanism, like the CPU in the cell. So it will control cell cycle, it will make the cell grow, it will give instruction to this, do that, don't do this. So that's the function of the nucleus. And when we will learn about amoeba and some other organism, what we call something nuclear division. When a nucleus one sorry, a cell division, there are two types of it. The nucleus will divide and the cytoplasm. If the nucleus doesn't divide, that cell cannot grow. So the nucleus controls almost everything in the cell. Is your question answered very well? Okay, she's off. Okay, so I'm sure that answers a good.
Yes, sir.
You are
Thank
very
you.
welcome. Please, we have just few minutes to end it. So that means that God willing, tomorrow, sorry, when Tuesday, I'm hoping we will have a meet. Even if we don't meet, I'll find a better way. We will meet on Tuesday. Yes, we will meet. Yeah, whether I travel or not, then I'll maybe use just my phone for the lesson. Harriet, your hand is up again. What is it now? Talk to us.
Okay, sir. The last
Okay,
question
it's even the last.
is
It's even the last. So finish it up.
we might have, can we have something like an organelle needs a substance, and then cytoplasm doesn't need that substance. But you see, the substance must pass through the cytoplasm before getting to that organelle. So would that substance having harm on the cytoplasm?
I get your question very well. Very, very brilliant question. So assuming, so now for instance, you are very high dehydrated. You need to take in water. You take in a little amount of water. All the cells also need water. What must happen? They have to share it equally among themselves. So the point is, and another thing is that, for instance, when we are doing connection to light supply and water to our homes, sometimes you are not connected to the main water tube. You are being joined to the one going to someone's house. So if you are joined to someone's line to the house, it means that you are sharing with them their power. So if they are using high rated voltage appliances, it means that you have unstable power connection in your home. If you are also joined to the pipeline leading to someone's house and they open their tap, it means you might not get a steady flow of water into your pipe. So that would be the hindrances. Or sometimes once you open your tap and they open theirs, the pressure from your pipeline will be reduced. That is some of the side effects. So if two of cytoplasm need water and is deficient, the others will also get, but it will not be as much as they need. So if it is one, because they are still embedded, so it's like you are washing, you have a little perfume or after wash in the container. So the water itself is having the afterwash, but if you put your clothes over there, you see that a little bit of it, of those stain and flavor, or some, how you call it, right? Is it the flavor or the scent will now enter your dress, but as if it was immersed in it. Most of the flavor and the scent could have entered all your dresses in that container of water that you are using for washing. So yes, it will be restricted. They will not get enough to be limited. But so far as they are still embedded in the cytoplasm, they will get very little to no based on the amount of water or resources inside the cytoplasm. Harold, are you okay?
Yes, please.
It's 9 o'clock on the dot. Let me just listen to Esther Kumache, then we are done. If you have any other question, you can post it on the WhatsApp page. I will address it tomorrow when the line is open. After 10, we will end it over there so that you will not say that I'm on the WhatsApp page whilst we are doing something on TikTok because our parents have to monitor us and we don't have to be there around 10. So 9, we are about leaving. Harry, do you have a question? Just ask it and I'm done. Sorry, Esther Kumache. Esther, hurry up. It's 9 o'clock.
Sir, please, so if you don't understand anything, this one, the YouTube one, will you give them the link so that they
Yeah, 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 yeah. So I have old videos already. That one is animation. So I told you that it's very difficult. It takes a lot of time doing the animation. So I stopped it last year. So I will share the link with you. Lesson one, lesson two, lesson three. All that we taught you in school. I have an animated cartoon video. I will share it today or tomorrow to you. I should have shared it right now, but I don't want to be sharing it every now and then. So after tomorrow or Tuesday, I will share it with you people. I am trying to record this. If I get a good recording on my tablet, I've kept it somewhere. I will edit it, I'll post it there and share it so that anytime, any day, you guys can go and watch. I'm sure that answers your question, Esther.
yes, please.
So any other person too who can help record, you can do so. After doing that, you can chat with me. If you can't share it via WhatsApp, I will give you my Telegram line or Telegram. You can share it over there. I'll edit it. Then we can have it over there. So if someone can do it for us, fine. If not, I'll be trying my own means. I'm hoping today I'll go and my tablet has recorded it very well. Then I'll edit and post it on YouTube channel, share it with all of you. Frederica, yeah, you will a question. What is your issue, Frederica? Yeah.
I don't join and I don't know the WhatsApp group you are talking about.
Oh, okay, very good. If you have my number, is that Frederica in one of Science 10 or
Science 9.
Science now? Okay, that is you. I can hear the voice, but I've forgotten the other name. That's why I couldn't add your surname. So, Frederica, if you have my number, chat me. If you have any of your friends you don't have, so ask someone. Then, how did you get the Google Meet link to join?
I got it from Speech.
Very good. That same link, the WhatsApp group is there. The same, I don't know how she sent it to you. Read very well. There's a WhatsApp page at the top. So ask Stacy to give you the group link, or you can chat any one of us here that you know. You can chat our classroom. They will share the link with you. Okay, thank you very much. So, people, thank you very much for joining us for tonight's lesson. So, God willing, tomorrow we will talk about this. Before you leave, we will talk about ways substance can move in and out of a cell. There are two main ways: passive transport and active transport. So, God willing, on Tuesday we will talk about diffusion, which is an example of passive transport. Have a very good night. Thank you for joining us. So, stay on the group and let's keep on studying the other subjects as well. Bye bye to you all. See you Tuesday, same time, 7:30 on the dot. Bye bye to you all. So you can start leaving the page